share a story with you today. It is kind of a fish story. Like Mark Twain said, don't tell fish stories where people know you, and certainly don't tell them where they know the fish. So I hope many of you are now familiar with the Middle Tennessee Cumberland River system, but I just want you to take note of the picture of the waterfall here. That'll be part of the story as it unfolds. As a result of studying the fishes of the Caney Fork River system, I became interested in this idea of microendemism in vertebrates. Not where vertebrates have microendemic distributions like relictual distributions, but where small geographic range sizes are a product of the speciation process itself. And to date, the best examples of microendemism with regard to this being a byproduct of speciation in vertebrates are in the tropics. So we have the Cryptotriton radiation along with salamanders in Central America, and of course the famous mouse lemurs in Madagascar, particularly the mouse lemurs in Madagascar, where these microendemic species, the number of species has inflated quite dramatically over the past 10 years. Now, in North America, many of you may not know, when it comes to freshwater fishes, North America is a biodiversity hotspot. Among temperate regions in the world, North America has many more species than all these other continental regions. And for example, the great state of Tennessee has more species of freshwater fish than all of Europe. However, that diversity of fishes in North America is not evenly distributed. You can see here with this heat map, there's a concentration of the biodiversity in the eastern part of the continent, and that diversity and endemism is concentrated in a set of fragmented highland areas called the Central Highlands, which in the west contains the Ouachita and Ozark Mountains, and even the hill country of Texas to some extent. Then in the east, the Cumberland Plateau and the Appalachian Mountains. Now, my taxonomic expertise focuses on just one clade of freshwater fishes in North America, clade darters. However, the patterns we see in darters are replicated across the other lineages that comprise this biodiverse freshwater fish fauna in eastern North America. There are about 250 species of darters, and they basically come in two flavors. If they're benthic in habitat, the males often have this bright, showy nuptial coloration, shown here with Ethiosmus sumatrum, or if they occupy what are called hyperbenthic habitats, that is water column habitats near the bottom, they tend to be more streamlined and lack coloration, such as here for Microsatellite. Now, the mechanism that people have proposed to explain the rich biodiversity in eastern North America, first articulated by Rick Maiden in the 1980s, is the Central Highlands vicariance hypothesis. And this has actually made the textbooks. This is taken from a textbook which I believe Dave Hillis is the co-author. It's a textbook we use at Yale in our intro biology sequence. The idea is that prior to Pliocene and Pleistocene glaciation, you had an extensive highland area that was geologically and climatically stable. You had a species-rich fauna that occupied that highland area, and that those highland areas were then bisected by the onset of glaciation. And we see remnants of this, or at least the biogeographic patterns that were driving this hypothesis, is this uncanny but yet replicated geographic pattern of distribution. That is where you have sister lineages. Here are 3 sister lineage clades of darters, where you have an Ozark species and then an Appalachian Cumberland Plateau analog. Now, not just darters show this pattern, several minnow species, and indeed even some sculpins show this disjunct geographic distribution, but indeed these are all sister species. So work that we've been doing over the last decade or so has actually shown that diversification between the highland areas doesn't really explain the biodiversity-rich area of eastern North America. And in fact, there's quite a bit of speciation within these highland areas. So what we're looking at here is a map of the Cumberland River system, starting up in Kentucky, going through, cutting through Tennessee, and dumping into the Ohio River back in Kentucky. And so here's a clade of Nothenotus darters, 3 different species, all of which they form a clade. And so what we're seeing within highland speciation is just as important, if not even more important, than speciation between highland areas. So that takes me to one of my favorite places on the planet, is the Caney Fork River system, which we're looking at Tennessee here. So if you drive on Interstate 40, going from Nashville to Knoxville or Knoxville to Nashville, you'll cross the Caney Fork River system 4 times. And basically, it looks like an inverted hand. It sits on the Cumberland Plateau, and where that red line is where that big waterfall is in the tidal slide. So the river comes off the plateau and dumps into what's called the Nashville Basin, a depression area that basically contains the city of Nashville and surrounding areas. Now this is a very small watershed. Drainage area is only about 4,250 square kilometers. Sounds like a big number, but in terms of drainage watersheds, that's actually pretty small. Now, interestingly, there are 5 endemic darter species to the Caney Fork. And these are not like oversplit molecular species that we will talk about later. I'm not saying they're oversplit, but these are species that were all established. One at least was utilizing molecular phylogenetics, but they're all phenotypically divergent from their sister species or sister lineage, and none of these species are closely related to each other than the fact that they're darters. And if we look at the geographic distribution within the Caney Fork system, 3 of the species, Ethiosma etneri, Ethiosma basileri, and Nothonotus starnzi, are found throughout the Caney Fork River system. Ethiosma forbesi is restricted to the western part of the Caney Fork, and Ethiosma acatulo is more restricted to the eastern part of the Caney Fork, but indeed gets into one of the western tributaries, the Collins River. So what I'm interested in doing is assessing genetic variation among these species, and even asking questions such as, do they share common divergence times between their most recent common ancestor and their sister lineages? Then determine if any of the Caney Fork species are showing diversification, speciation within the system. So what I've done here is utilizing a complete sample time-calibrated phylogeny of darters. Here we have the Bayesian divergence time estimate posterior, mean posterior in millions of years, and the whisker shows the credible interval around that age estimate. What we see is that 3 of the species, Ethiosma etneri, Ethiosma basileri, Ethiosma acatulo, have a very similar divergence time, somewhere between 10 and 12 million years between them and their sister lineage. Ethiosma forbesi has an intermediate divergence time, and Nothonotus starnzi is actually quite young. So we seem to have 3 different sets of divergence times that explain those 5 endemic species. Now, if we look at genetic variation among those 3 oldest species, here we have Ethiosma etneri and Ethiosma acatulo. What you can see, these mitochondrial DNA haplotype networks, there's really no association between geography and the distribution of haplotypes. There's a little bit of a trend in Ethiosma acatulo between an east-west divergence. Acatulo, if you remember, was in one western tributary, but not really. However, Ethiosma basileri shows really dramatic geographic variation with regard to mitochondrial DNA haplotypes. Indeed, essentially, mitochondrial haplotypes are not shared among any of the 7 tributaries of the Caney Fork, of which basileri occupies. They're color-coded here on the map. By the way, this map represents all of the known historical locations for Ethiosma basileri. And if we do a base time calibrated phylogeny, mitochondrial gene tree, we find we resolve 7 lineages with a common ancestral age of about 5.5 million years. And the youngest divergence is 1.2 million years. However, there appears to be, as you'll see when we look at the Radseek data and exonic star-based species tree inferences, it looks there's a mitochondrial regression event between the Barren Fork, which is in green here, and Hickory Creek in purple. So a star-based species tree inferred from 13 exons of Sanger legacy genes essentially resolves a strongly supported species tree that gives us a nice split between the eastern populations and the western populations, and again is incongruent with the mitochondrial gene tree with regard to Barren Fork and Hickory Creek. We went ahead and collected microsatellite data from microsatellite loci designed specifically for Ethiosma basileri, and essentially what we find using a Bayesian structuring approach, we get an optimal K of 7 in structure. That is, each tributary has its own kind of genetic cluster, and there's very little admixture. Assignment probabilities are high, essentially, for the case. So we went ahead and gathered a Radseek data set. At this point, I kind of call it a weather balloon, test balloon, of about 33,000 loci. We get a fairly well-resolved phylogeny that is essentially congruent with our star-based species tree estimate. So how many species are masquerading as Ethiosma basileri? Well, if we look at the mitochondrial gene tree, taking a VGMYC approach, it says 6 species, but remember we have this potential of mitochondrial regression between those 2 lineages. If we look at the 13 nuclear genes and using IBP, we find that 13 nuclear genes, we get very strong posteriors for a 7-species delimitation scenario. If we add mitochondrial DNA, despite that pattern of introgression, we still have a very high support for a 7-species scenario. However, there's this issue, are these cryptic species or not, and particularly with among many of my ichthyological colleagues, particularly North American ichthyologists, they find molecular delimited species problematic. So we went ahead and collected a classic Baristic data set. These are essentially traits that have been used since the 1820s, still used in the early 21st century to delimit and describe species of fish. Fish have things you can count, like scales and fin elements. And what we found is that for any given lineage of Ethiopsum basilaria, there are at least 2 meristic traits that distinguish them. And don't take my word for it, I'll just show you 2 traits. One here, we're looking at the frequency distribution of the number of pectoral fin rays. You can see Barren Fork, which is actually the type locality for Ethiopsum basilaria, has 13, everyone else has 12. Some of these meristic traits show a even deeper biogeographic pattern. So if we look at the number of dorsal fin spines, we have modally 8 in the west, modally 9 in the east. We can take this data and do a canonical discriminant analysis, and basically the model that we use the data as a training data, it has a correct classification 70.9% of the time, which is actually really high. Just given expected by chance alone, we would expect the model to perform at 14.7% accuracy. Now, what we did is, the nice thing about IBPP is that you can incorporate phenotype in your species delimitation. So using the guide tree that we essentially inferred from the Radseek data, we just looked at meristic traits only. So this is phenotype only. And again, we have fairly moderate support for the 7-species scenario. So basically, there are 6 undescribed species that are currently referred to Ethiopsum basilaria. Some actually have some compelling pigmentation differences. Maybe you have to have a sharp eye, but you'll notice this guy has a little dash of jet black in his tail. This fellow does not. This is something that's consistent among specimens in these populations that we're delimiting. Some of these species have the smallest range sizes of any vertebrates in eastern North America. Very, very small range sizes. And the descriptions are in progress. But I think more importantly, what does this tell us about in terms of the evolutionary biology of the system? Well, clearly in the KD4, there's kind of an idiosyncratic history among these 5 lineages. So we can reject like a singular event, like a singular vicariant event that explains this. And what I find interesting is that the oldest endemic lineages in the KD4 all appear relictual in the sense that they are sister lineages to much more species-rich clades. And the 2 younger species are phylogenetically nested in other clades. So it's clear that areas of eastern North America are acting as kind of biodiversity museums, preserving biodiversity, but these same areas are also acting as cradles where this biodiversity is generated. And I think this will involve the size of the area and the demography of the species. And in the case of basilaria, its diversification in the KD4 is not likely classic vicariance, but I'm assuming it's more dispersal-mediated geographic isolation. With that, I'll be happy to take any questions. Thank you.